Today, Benjamin Good from TSRI will talk about three tools that aim to capture, share, and apply community knowledge. Ben will be giving updates on the GeneWiki project and then talk about Branch, which explores biological data using decision trees, and also Knowledge.bio, which provides access to a network of interconnected concepts mined from more than 20 million PubMed uh, abstracts. Without further ado, allow me to introduce Ben.
Hi everyone. Can you hear me okay? Just
Yeah.
last check? Yeah. Okay.
Yes.
Great, thank you. Yes, thanks. Just want to say why I have the sign up there. Everything I'll be talking about is very much a work in progress. This is the first time I'm presenting most of this, and so bear with me. Everything should be working though, so this is a good time to give this talk because once you go off to the URLs that I'll show you, all the tools should be functional to you, and it's a great time for us to gather some feedback from you about how to take them to the next step. The slides should be posted from Vincent. They're also posted to Twitter as of this morning at BeGood. I'll get this to move. There we go. So, like you said, I've got three parts to this talk, not super interconnected, so that's why I have an outline here. I'm going to give you a very quick update about the GeneWiki project. The previous presentation is really a much more thorough that I gave in April, which you can get through our channel, a much more thorough introduction to the GeneWiki project. This one I'm just going to tell you where we are right now after that one, and then I'll talk about these two new applications being developed in our laboratory. So, for those of you who don't know about it, the GeneWiki is a wiki that happens to exist on Wikipedia. And when we say GeneWiki, we're talking about all the articles there that are about human genes. Our lab's responsibility is to provide software for creating stub articles and maintaining them. It does this through control of these sort of structured data items that you can see, things about the protein structure. Importantly, links off to different databases and so forth. And so basically we help generate new pages like this, and we make sure they stay up to date and high quality, at least in that area. The GeneWiki project is migrating work to something called Wikidata. So Wikidata is a new database that's going to be deriving content on Wikipedia. So all of that structured data that you see here is now going to be moved into the Wikidata database. As you can see, there's items in there about genes with structured information about them that you can query just like any other database, which is a major step forward in terms of how the MediaWiki family manages data like this. So what we're doing right now, we're taking structured data, as we said before, from various databases, mainly mediated at the moment from mygene.info, and moving it into Wikidata. So that's where we are right now. We're writing code to do that. The next phase, which will be happening soon and we're already experimenting with, is getting that data into Wikipedia. Following that, during the course of the next year or so, we'll be seeing what we can do, what else we can do with it. So can we get structured annotations back out from the Wikipedia contributors? And one of the great things about this new model is that data could actually serve any application you could imagine. It's an open database, so Wikipedia is just one of them. And so we're excited about what may come out over here. So, status update on this project. We have entries about genes, diseases, and as soon as they let us run our script, we'll have a whole set of information in there about drugs. The set of drugs will be coming from Drug Bank and will be drugs that are approved by the FDA as our starting point. We do have demonstrations of incorporating this content in Wikipedia, although, like I said, that hasn't gone large scale yet. The technology is there now. And so we've been slowed a little bit in this project by Wikidata governance policies. What that means is that one of the great benefits of working with Wikipedia and Wikidata is that you have these large communities of people that you work with that can help you do good work. spread the labor around, but it also means that you have to convince them that what you want to do is what they want to do, and sometimes that can be a slow process. I mention that because anyone can help, including anyone on the call. If you want to see what we're doing on Wikidata in particular, go to that URL, and we keep track of all the conversations that are going on there. So, for example, when you go there and you look at all these links, will go to take you to a place where you can see what people are talking about and voice your support or disagreement for what we're trying to do. And so the more people that are involved, that are really engaged and knowledgeable about this stuff, the better this project is going to be. So I'd really encourage and welcome anyone that wants to join us there. So that's all I want to say about the GeneWiki project for right now. I'm going to just pause for a second just to see if there are any questions about that.
This is Peipei.
Mm-hmm.
I think it's a very important project. Could you share with us more insights on who's making the decision whether or not the project will go forward? Why would rally public support be important?
Okay, so just let me repeat because it's a little hard to hear you. So you're asking who makes decisions about the bots and why it would be important to rally public support. So basically, the organization of Wikidata and Wikipedia itself is that basically anyone that comes in can make contributions. Okay, so we can do things, but when you're writing bots, you're writing code that will edit thousands or hundreds of thousands of pages. Some form of governance is needed so that things don't go really badly if someone doesn't know what they're doing when they get in there. And so what they do is they basically have kind of elected, trusted authority people that, like for example, in Wikipedia they have people that control the article about Barack Obama and make sure that it doesn't really go haywire. And so it's the same kind of thing in Wikidata. There are people that manage the bot tokens, the people that have access to make mass edits in there. And so, and that's a good thing, okay? Because it means that you stop people from spamming it and so forth. And all it really means is that we need to be real careful and go through the process. So what happened in over the summer was that one of our developers made a mistake writing his bot code and ended up creating a really large number of duplicate entries in their database, which no one wants, and so they put the button that says stop. So that made it so that we could not run that bot anymore. And this launched this process where we had to get it reapproved and go demonstrate that we had fixed it and so forth. And all that is working fine, and we're not really worried about it ultimately achieving, getting it back, because we work closely with everyone there. They basically trust us, but the whole process goes faster when you have more voices on your side. And so when they put up a page, like you'll see in those links that I have back there, it's an open discussion page. And if you say, okay, I'm asking for permission to do something, and you have people come in there and you have 5 people say, yes, I support this, yes, I support this, yes, I support this, that will make it go faster than if you put it up there and no one says anything, which is kind of what happened during the summer.
Sure.
Does that make sense?
Now it does. Now it's clear then how we could have people join in the conversation to support it. That's excellent. Thank you.
Okay, no problem. Okay, so so that's the GeneWiki project and where we are right now. And now for something completely new that probably no one's seen before, so which should be fun. So this was something that is called, for the moment, Knowledge.bio. So the idea here is to provide a concept-centric view of the scientific literature. That means that when you do a search, you search for a concept, interact with concepts, rather than searching for documents and long lists of documents. The main purpose for users is hypothesis generation. The two data sources at the moment that are driving this application are both mined from the full PubMed abstract corpus. There are about 70 million explicit semantic relations, or I'll call them triples. Those are coming from a tool called SEMREP from the National Library of Medicine UMLS team. And then we have about 200 million implicit gene disease associations. So when I say implicit, I'm saying that there's a candidate association, and we don't actually know what the property is that links that gene to that disease. But there's some indication that there's a relationship there. So if you go to Knowledge.bio, you'll land on a screen that will quickly look like this. You click the demo button. There's a lot going on here, so what I'm going to do in this talk is break it down piece by piece and try and go through a little, basically a walkthrough of how you might use it. So you're going to start when you come in here with this little search box, and you're going to search for a concept. So in this case, I'm searching for a gene, the CYP2R1 gene. Okay, that'll give you a little interface to choose from multiple choices in case you have multiple hits on it. You'll pick the choice, and then it'll show up like the screen is indicating there. And you'll be able to see all of the explicitly related concepts in this table here. And you'll see the subject is the gene, a bunch of different kinds of relationships, an object. The gene may show up in either the subject or the object location. And there's quite a bit of information that can come back. In this case, you'll see there's 67 results down there. And so we have this ability to filter that list down. So right now, it's being filtered by text, so you can actually filter on any of the text strings in the table. Underneath that, there's a semantic filter, so if you only want to see, for example, drugs related to that gene, you could use that semantic filter to do so. So this is the main kind of search interface in the application. So the other thing to point out here is that those little green buttons on the side with different numbers in them indicate how much evidence you have from the text to support that claim. So in that case, CYP2R1 affects eczema has one statement that supports it. And so when you click that button, you'll be moved down to the lower level of the application in a tab called Citation Evidence. So we came from that button, we moved down, Citation Evidence tab was opened, and then you can read the sentence here where that statement was extracted from. So CYP2R1 affects hydroxylation came from this little fragment of text right here, which you can decide whether or not you believe, basically. And if you click on it, it will open up the other tab here with the full abstract in it. If you click on that, then you can view all of the other relationships extracted from that abstract. And so this is how you sort of interact with the literature. So coming back, you would go back out to that original search query, and it starts off in explicit relations, and now I've just gone and selected the implicit relations tab here. Like I said, these are the ones where we have only links between genes and diseases, and basically a statistical measure of confidence that there's some relationship there, and that is indicated over here. So for the gene CYP2R1, we have a strong hit on this Smith-Lemli Opitz syndrome right there. Okay, all these are sorted by that, and you can see in many cases there are quite a number of potential entries there. So when you click on, you can click on that number, the score, and that will move you over to the next tab, which indicates to you why the concepts that indicates to you why that score is what it is. And so basically what happens is that it looks for co-occurrences between the original search term, so in this case CYP2R1, and other concepts around it, so like DHCR7. Even though CYP2R1 never co-occurs with Smith-Lemli Opitz in the same abstract, they both co-occur with these other concepts like DHCR7 independently. And that's how they draw those associations. There's a lot of math to that, which I'm not going to go into today, but that's how it works. And I'll go into another example that explains that a little more clearly, I think. Next. Okay, so coming back to the full screen of the application, you start there in that explicit relations tab, and you can run around and look at your data here, and in the implicit relations tab, you can look at the evidence view on the bottom. And what you have on the other side here is this network view, which will start empty, and then when you decide you believe in something, you can throw it in there, and it will basically be used as a way for you to keep notes in your discovery process and assemble these maps that you can view and interact with and share with your friends. And so this map view is composed using the cytoscape.js network building library. You can use its automatic layout features using that little green layout button. You can also drag things around, interact with it, zoom in, zoom out, and so forth, like you would in Cytoscape. Right now, this works. You can save the map as a local text file, so you can save it, email it to someone else, and then they can go and load it up over here. It's a little sort of early stages for that process, but that whole life cycle does work. That can be very useful, of course. Oh, just to point out, there is, if you want to print out what you have here, you can turn off the dark and it will turn into a black and white view that is much more suitable for printers. And so now I'll just go through a little workthrough of how you might use it. So you might say, okay, I want to discover something new, new candidate relation. And the question might start with, as I start here, what new diseases might be related to X? So in this case, the CYP2R1 gene. You do that search, you find, as I said before, you find this hit on SLO syndrome, and what you'll do is you'll actually grab that and drag it into the map area, and that will create this relationship. You'll see the gene, the disease, the number of the power of the association. So you just added that implicit prediction to your map. The next step would be to ask the question, how is CYP2R1 related to SLO syndrome? So why is that there? Like I said, you go over to the co-occurrence tab. See that first concept is very highly contributing, and again you'll grab that and add it to the map by dragging and dropping, and that will introduce these two new connections called mentioned with, because that's all the kind of relationship that we know about for those two things, and now you can really sort of see how things start to fit together from the implicit view. Next question is why, right? So now we want to start approaching a hypothesis. So do CYP2R1 and DHCR7 participate in a process that's related to SLO syndrome? So now I would go back into the explicit view and start building up content about those genes. So in this case, you'll see lists like this, like CYP2R1 affects hydroxylation and vitamin D metabolism. I'll grab that, I'll throw it in there. And then, as it turns out, if you go back and look at DHCR7, you'll find that it is also affecting vitamin D metabolism. And if you do a little more research from there, you'll find that this is actually an important part of the pathogenesis of Smith-Lemli Opitz syndrome. So that's a good example of how you could use the system to discover a potential hypothesis and then move towards strengthening it and digging out through the literature in ways that you would have much more difficulty, we think, doing just directly off of PubMed. So that's the idea with this application. To tell you, I believe I'm probably the only user so far that we have, but it's actually quite fun once you start getting into this. It's a really interesting way to rapidly find connections between things. So I encourage you to go in there, http Knowledge.bio, and have a play with it, and let us know what you think and see how we can make it better for you. The immediate next steps that we have for this application are really to work on enhancing our community sharing capabilities. And the way we're going to do that is to integrate the application with something from the Cytoscape consortium called Index, indexbio.org, whose entire purpose in its application life is to share networks. And so the networks created in our application will be deposited in and managed by Index, and thus integrated with the whole Cytoscape universe of other networks. That will mean that you can create networks here, go to your computer and load them up in Cytoscape, and potentially vice versa. So I think that will be a very powerful connection to make for this guy. The other thing that we're very interested in doing, and is actually some of the original inspiration for building this application, is to capture information from the users themselves. So to allow the user actions in the system to feed back into the underlying natural language processing systems to improve them. So when you get in there, you'll notice there are definitely errors in the triples that are extracted from sentences. It would be nice to be able to capture that knowledge from you, which will be very obvious, and throw that back into the teams that build the NLP systems and thus improve them. There's also different kinds of knowledge you could capture. So some of them may be correct, but not interesting. And that the idea of interestingness is something that is like entirely human and very difficult to model. And it's also something we could capture through the system. And the other thing, of course, there's all kinds of other data that we might want to add to this system, and we'll do this basically as resources allow. So, for example, I'd really like to add gene ontology annotations into it. So I'm going to pause again before moving on to the next one. Are there any questions about Knowledge.bio?
Hi.
Hi, Benjamin.
Hi, Ben.
This is... Oh.
Oh, sorry.
No, you go first. I'll go second.
Yeah, this is Brian. I was just curious. So, I know you have like the relationships of facts is located in or is a part of or something like that. How did you define all those relationships?
Sorry, I missed the first part of your question. It's
yeah, in the triples, how do you define the relationships?
How do you define the relationships in the triples?
Yeah, because I know there's a limited number of them. So did you guys explicitly, determine that there would be 20 relationships or something?
Okay, sure. So that is really entirely coming out of this tool called SEMREP, S-E-M-R-E-P, from the UMLS team at NIH. And so that's really, they basically do as many relationships as they think they can do well. There's no, and they focus on relationships that they think are going to be useful. And so the total set of them that they cover is there's about 50 of them, which is a pretty large set. It's not bad. There's a lot of information, little different kinds of relations in there. I could also tell you that they vary dramatically in terms of how good they are at getting one of those relationships right. So one of the more interesting relations, for example, is causes, of course, but I would say a large percentage of the causal relations that are in there are probably not right. They indicate some relationship, but cause is probably too strong to say what it is that they indicate. But the best place to learn more about that is if you just Google for SEMREP, and you'll land on their stuff. Okay.
Okay, and then one other question. So for the graphs, I know you say you drag a term onto the graph and then it shows the relationship. Do you have a way to automatically populate the graphs? Like if I'm just interested in a particular gene, but I don't know where to go with it, I could just see, things that have a close semantic relationship to a particular gene would automatically be populated to show the nearby neighbors.
Well, so that was the decision at the beginning of this. At the moment, the answer is no, and the reason is that I don't want to throw someone into a giant hairball,
Mm-hmm.
And unless I have a really good way of knowing what it is that they're after, I think it's going to be better for them to actually interact with that searchable, filterable list than it is to interact with it directly in the network. So that's, a lot of times people kind of just go immediately to the network view, and it's my opinion that it's better to use that as, like I said, as a kind of a note-taking device and interact with textual searches and filtering and sorting on the other side. That being said, one of the things that we are interested in doing is adding a 2-node pathfinding component to this. So given that you have two things that you're interested in, find potential connections between them. That will constrain the sort of the hairball generation process a bit, and it's something that people really clearly want. I know that they want, and so we're looking into ways of making that happen. Most likely, we're going to use something called Neo4j to do that and to add that kind of the kind of functions that you're talking about. But I go into that hesitantly because I'm just not a big fan of throwing people into hairballs. Okay?
Yeah, thank you.
Sure. Was there one more?
Hi, Ben. This is Sarah Scruggs. Yep. Can you hear me?
Yep.
Hello?
Hello.
Okay, awesome. Great talk. This is really very interesting. I'm still sort of processing it, but I did have a couple initial questions. The first one is, I'm trying to think about how this sort of jives with Wikidata. They sort of seem complementary, and I'm just trying to see sort of how your goals align.
Okay, yeah, great question. Something that we should flesh out more. So, number one way they align would be Wikidata may be one source of knowledge to tap into for applications like this. Okay, like I mentioned, that's one of the goals of the Wikidata project is to support not just Wikipedia but also other applications in this area. So that may be one way that we can actually simplify the development of this, so use it as basically a data warehousing system. The other side of this is what we want to do, and everything that we're working on is capture information from users. And so we can do that through Wikidata, which could feed into these systems, like I said, but we can also potentially do it from the applications themselves. So when people find a relationship in Knowledge.bio that isn't correct, if that came from Wikidata, that application could then feed that information from the user back into it and thus make it better. So that's kind of how I see things fitting together, both on the sort of, pulling content out and then getting feedback from users. And this is just another way of getting more, and particularly more expert users into it.
Sure, great. My second question is, I'm sort of thinking about this from a cardiovascular standpoint. Is there a way you can sort of set filters for articles in the PubMed database? Do you see what I mean? So you're only incorporating information from, say, your topic of focus, like within the cardiovascular field, because gene X could have, mutations in gene X could be associated with, several different types of diseases in different, organs. Do you see what I'm getting at?
I do. So you're suggesting an article, like a journal level filter for it, in addition to.
So I'm sorry. Go ahead. Yeah, that's
sorry,
sort of.
The end there.
No, that's sort of what I'm thinking. Because if I had my note screen, I would want to be mapping out these pathways of disease within sort of the cardiovascular system, or probably just within the cardiac system. So I'm wondering if there's a way you can sort of hone it into a particular.
Right, like a keyword search, Sarah. This is Carol Watson.
Yeah. I don't know if this is something I should think about further, but I was just thinking like, oh well, in writing these GeneWiki pages, some of these genes are, cardiac specific, others are certainly not, and I'm just wondering, it would be of the most use to me in particular if I could sort of focus on the cardiac system.
Well, okay, so sorry, every time someone starts talking, it like cuts out for a few seconds in the beginning, but I think I know what you're asking. My first response is I'd be really curious for you to go there and try it and see how it feels to you, and see if that you really do feel like you're overwhelmed with data that's not relevant, or whether you can use it as it stands to do what you want to do. Because I could imagine putting in sort of additional filters on the data that would allow you to sort of break it down. But on the other side, the whole, one of the real points of this application is to bring connections to your eyes that you wouldn't normally see because you wouldn't normally be looking at them. So there may be interesting things that you find, interesting genes, for example, that you find that are relevant for reasons that you might not have thought about before. And so, I mean, there's definitely a tension between, providing you with too much, kind of overwhelming you, but we do want to really push on the idea of having it kind of at your fingertips, just one step away. But I mean, I definitely hear what you're saying, and the way of achieving that is to be able to have access to the full content, but be able to filter it the way you want it. And so.
Yeah, I'll go play with it. You're absolutely right. Shouldn't criticize until I play with it. Thank you.
No, 'cause I mean that would be great feedback. I'd really like to hear
Yeah.
how that works out, and because I do think we could add things that would move things in that direction if that was really worthwhile.
Okay. Very good. Thanks.
Sure. Okay, unless there's any other questions, I will move on to the last part of the talk. And if there are, I can also see the chat window, so you can chat them in there. Okay, moving on to something completely different. I'm going to have to step back and talk about the beginning of this. So this is actually a project that had its inception, theoretically, about a couple years ago. And the problem that we were working on at the time, and still is a very kind of core problem in bioinformatics, it's that of inferring class predictors. So the problem we were working on then was breast cancer prognosis. So what we wanted to do, and what people have been working on in bioinformatics, since basically the beginning of bioinformatics, is to take a molecular sample, for example, gene expression data, and use it to make a prediction about 10-year survival for breast cancer, as that knowledge can be used to influence people's therapy. And to do that, we of course take that question, we go off and accumulate these large data sets that used to look like this good old-fashioned gene expression data set. We feed those into large numbers of computers and statistical algorithms, and we hope we find stable patterns that we can use to make predictions on new sample. Okay. Sort of oversimplifying that middle process of finding the patterns, there are two key steps. The first step is to pick your features and pick your genes. The next step is to infer a predictor using a variety of different methodologies, such as decision trees or support vector machines or neural networks or etc. And so in the beginning, what we focused on is this, on picking the gene. So out of many thousands of genes, which small set works together the best? The best case scenario is you can find a small panel that you can use to make a test with. The problem, of course, is anyone that's worked in this area will be very familiar with, is that everyone that does this ends up picking a different set of genes. So Veridex, Oncotype DX, Mountain are all commercial tests built for solving this problem. You'll notice that there's very little overlap between them. This RFRS is a recently published method to do the same thing. Also, again, very little overlap between them. Different data sets, different methods end up picking different genes given the scale of the data that they're looking at. And that sort of is unsatisfying for many reasons, the most important of which is what we refer to as the validation gap. And this is the problem in that when you're doing your work, obviously on your training data, things look better than they do on the test data that you have, but even worse is the validation data set that comes down the road later. Getting across that gap is one of the primary challenges of this whole field. And so one of the ways that people try and do a better job at this is to add prior knowledge to the discovery algorithm. So the idea here is that when you come in and you're looking at this data set, we actually know quite a bit about a lot of those genes and so forth and how they relate to one another. And maybe we can use that to constrain our search for the right patterns to do a better job in building a more stable, more reliable predictor. And so quickly, one nice example of this from the Eidecker lab at UCSD was something called network-guided forests. And their idea here is they used the protein interaction network to select the sets of genes that would be used to create the trees in the forest. And it turned out that resulted in a more stable, more accurate, only slightly more accurate system. The problem, of course, is that, as you will notice, that most knowledge is not structured. Can't give a talk without showing this slide about the growth of PubMed. We're now, over 100 publications an hour, more than 1 million a year, nearly 200,000 linked specifically to breast cancer. And so not everything lives in protein interaction networks. And so the question is, how can we use unstructured knowledge to improve predictors? What we need is a distributed network of intelligent systems that are good at reading and hypothesizing. And so, of course, being a crowdsourcing lab, this is what we think about as you and, of course, all of your friends. And so I won't go into this too much. You can read all about it. You can still play it. Our first attempt at this was a game called the cure, you can go to genegames.org/cure and play it, and you can read a paper about all the results. The only thing I want to mention about it is that people interacted with it, they liked it, but what they really liked was the little tree on the right there, and they wanted control over it, which they didn't have in the game. And so from that, we got to branch. So if you go to biobranch.org, you can get in there and play with this thing right now. It's live on the web. It's developed by a guy who was an undergraduate research student with us last year and is now just starting his PhD, and maybe this will turn into his whole PhD thesis if people like it. And so you go in there, and so I'll just say the purpose of Branch is to provide an easy visual way for non-programmers to use very large data sets to answer questions. Okay, that is really what we're after with this particular thing. It's moved on from game to tool, although it may turn into a game again at some point in the future. And as a byproduct of that, we were hoping to construct large libraries of manually crafted predictive models, which we could then use downstream to generate ensemble predictors that incorporate the knowledge of the users. And that's kind of stuff in the future. Right now, it's really focused on being a tool for users to do their work with. And so I'm going to do like I did with Knowledge.bio. I'm going to try and give you a kind of a walkthrough of how you might use the tool and all the different pieces that it has. And so once you log into it, the first place you'll land is the dataset library. So right now we've got these 4 datasets loaded. We're hoping to have more over time. But we'll go in there and we'll pick one. So I'll pick the breast cancer survival prediction dataset. This is actually from the Griffith paper that I cited earlier. This is the same structure of data that I mentioned before. So in addition to gene expression data, it has clinical features of the patients. And so the goal being to make a predictor for predicting no relapse or relapse from breast cancer. So you click the Use button, you go in there, and in this case we have both a training set and a testing set. So I picked the test set to use to evaluate my model. If I didn't have that, I could evaluate directly on the training set, or I could do a percentage split. So I could cut the data up into, you know, a 60% training and 40% testing, for example, right there. And then you will land in here. This is the kind of primary screen of the application, and where you're going to build your decision tree to use to make predictions about that data set. And so I'm going to piece this apart again bit by bit to show you how it works. It's actually fairly feature-rich, so bear with me. So the core of this thing is the split node builder, what you see right here. From there, you'll see lots of different ways to compose different kinds of split nodes in your decision tree. Here's an example of a split node. So one feature in the data set is the age of the patient where the sample came from. And what this thing is doing is saying, okay, we're just going to start here with all of our data. We're going to cut it at, in this case, at the age of 34.5 and say anyone that is younger than that, we're going to predict relapse. Anyone that is older than that, we're going to predict no relapse. And the colored squares give you an indication of how much data is going where, along with the purity of the node in the tree. So you have an idea that the goal here is to divide them all up so that you'll see all red and all blue. If you click on the class distribution button there underneath the box, you'll be able to see all the gene expression values and basically a histogram of all the gene expression values and where the split point has been set. If you care, you can grab that thing that says drag and move it around, and you'll see the data redistributing according to where you set it. It's set automatically using statistics at the moment. So that was an example of a single feature split on a clinical feature. So this thing is very much set up to work on genes as well. So we have lots of different search functions, for example, built in for genes. If you select genes, you go into the box and you start typing, it will do a search over mygene.info for the gene name, annotation on the genes, and so forth. So that's one way you can find things of interest that you might want to look at in the tree. You can also use a statistical ranking of the genes that will basically give you an idea. If you don't know what gene you want to look at in that context, it'll basically make suggestions for you that are can be very useful. And so that's single feature splits. This is kind of the simplest way to go about doing this. This is the way that you would typically go about building a decision tree if you were a decision tree algorithm. In addition to this, we'll move over to the next button there, and this is a way that you can custom build feature combinations. So you can manually compose a linear combination, so an equation of other features that will create a new independent feature that you can use as a split node. So if you have, so for example, you have a feature about the expression of BRCA2, you have another one about TOP2B, another gene. You can use this tool. You basically find the build custom feature button to do this, and add them together. You can create equation, and that turns them into one view. Hello? Sorry, somebody was talking. Okay, I will continue. And so, you know, it may seem like an odd thing to do, but we put it in there because we wanted to be able to reproduce this 21 gene signature from the Oncotype DX test as it was published in the literature. So we wanted to have that capacity in the tool. And based on that tool, we do. So what that signature is a combination of genes, basically a linear combination of the expression values of these different genes grouped into these different categories. And I won't go into the long details in the slide here, but the essence of it is that using the tool, we can come up with their recurrence score. So that recurrence score, which is this combination of all these different gene signals, can now be used as a node in our breast cancer decision trees, which is somewhat complicated to use, but it's actually a very powerful feature. Now, coming back to this, if you don't actually know the numbers that you want to use in an equation to build a particular feature, you might want to use a machine learning system to come up with those numbers yourself. And so we have the ability to do that in here. So down here you click Build Classifier, and you would launch an interface where you can create a classifier. You basically pick whatever features you want, genes and clinical variables and so forth. You pick the model that you want, naive Bayes, support vector machine, a decision tree itself. It would actually train the model, and then you could come back here and use it. Anyone else could use it, and it would act just like anything else in the tree. So data would come into it, it would execute the model and make its predictions, and then run the data down either of the binary predictions of the tree. So I think that's actually a very powerful little piece of logic we have in here. The trick is making it easy enough to use. So the other thing, of course, along the same lines, you can use a previously constructed tree as a node. So someone may have built a tree that makes predictions based on a certain set of genes. Let's say it's someone's focused a decision tree on the immune-related genes. You could grab that tree, throw it in as a node in your tree, a single node, take its predictions, and then work from them to stratify them yourself. And so again, it works exactly the same way as the other one. And the last way to build a split here is visually. And so this is something where you don't actually have to have a lot of prior knowledge to start doing something that may or may not be useful. I'll show you how this works. So you click the plot button, and you'll go into an interface that will look like this. First thing that you'll do is pick a feature that's going to compose the y-axis. The next thing to do is to pick a feature that will compose the x-axis. So in this case, I picked TLRR5 and CHST15. After that, there's a button over here that will say plot. It will plot all of the samples for you in this area. It will color them based on their class, and then you will be able to draw a polygon that represents your decision boundary. Okay, so in this case I circled that area so that the tree is going to basically make a prediction that if things lie in that area, you will go one way, you'll predict the CBF0 class in this case, otherwise you'll go the other way. You'll see over here in the selection statistics area training data that you're seeing. So what you see here is only what you've selected as training data. You do not get to see the test data. When you go in next, you say, okay, build split. You'll add it to the tree, and you'll come back, and you'll be able to see how well it actually does on your test data. That this pattern has been demonstrated to be useful in some cases in the past, and so we're really curious to see what people can do with it. There's certainly bad things you can do with it, or you could. So, for example, one of the best things you might want to use is teach your students about what overfitting means in terms of creating decision boundaries. So you can draw a batband in there, or you can circle all of your samples. Once you actually test that, you'll find that it's actually a terrible predictive model. And if nothing else, it's good for teaching students about that, making it real. So those are all the different ways of creating the decision tree nodes. The other panel here on the right is where you see how good you're doing, basically. Breaking that apart, you can view the training and the test sets that are being used at the moment in your data. So here I'm using a separate training and testing set. The accuracy of whatever tree I'm looking at is 67%. The area under the curve is 0.65. You can look at the ROC curve, and of course you can view the confusion matrix that underlies the accuracy measure. Once you're done building a tree, or you want to start out, so navigation panel. So that was the tree building interface. The other parts of the application are this button here, which will clean out your current tree and allow you to build a new one. The other very important button there is to save your tree. Once you click that button, it will go into your tree collection. You'll have the option of keeping your tree private to you or saving it in a communal area where everyone can see it and contribute to it. So this is the tree collection area, and so right now if you click on tree collection, you'll be looking at just your trees. So these are the trees by Benjamin. If you click on community, you'll be seeing all the trees that have been shared by the public. This area has a search feature, so you can search by text across all of the comments and all of the names of the genes and so forth that used in the system. It actually works very well. This column of the data will give you a preview of what the tree looks like. You can sort by all of these things, so you can find the most recently created trees by your friends, and so forth. Key here is that you have a button way over here on the right that will allow you to open and edit your previously created trees or any of the open trees. You click that button, you'll land back into the tree building screen and be able to work directly from that tree and then save it off as a new one. Sort of like a little bit of GitHub in the middle of tree building world. One of the things that it will do, it will track who created or contributed to each node. So if you click on the blue one, you'll see the developer, Karthik. You click on the orange one here, you would see me as I added that node to that tree. And that is a way we could imagine of distributing credit back to the people that contributed to these models. So the next steps here for this application, as I said, these are all kinds of work in progress. More user testing, we want to keep smoothing it out. So if anyone was interested in using this, we'd be pretty excited to hear from them to get some feedback about it. Along those lines, obviously this thing isn't super useful unless you're interested in the data. So one of the important things that we need to work on is figure out ways to get more data into it. There are both sort of technical challenges and decisions to make about how to do that going forward. And we hope, the goal again here is that if we do a good job with those two, we'll end up with getting lots of people to use it. And that's where, to me, the interesting potential of this application begins to arise. And so can expert users interact with a system like this to build better models? It's an open question that's very dependent on who the expert is, how good the interface is, and what kind of data they're looking at. And if they can, maybe we can do a better job at getting across this gap. So finishing up now, for more information about Biobranch, there are screencasts that you can use to walk through all the other features that I didn't get into in depth there at tinyurl.com/branchcast. All of these projects that I talked about are open source, and we would really be excited about anyone that wanted to take it build something new with it, or contribute back to it. So Biobranch is under the SULAB repository. Knowledge.bio is under this one. It's being developed by a contractor at Star Informatics under GBK, which stands for Garden of Biological Knowledge. All of the Wikidata and Wikipedia stuff is going to be living here under SULAB Wikidata bots. You can go see how to talk to, read and write from Wikidata there. And again, we would welcome any contributions. And with that, I would say thank you to Andrew, the Prince of Crowdsourcing, who organizes all of this for our lab and gets us funded. And to Karthik, who built and continues to build the branch application, as well as Vaishak, who is a summer student with us that also helped. Richard Bruskowicz is our developer from Star Informatics, who's building Knowledge.bio. The implicitome data is coming from Leiden University Medical Center. Our contact there is Christina Hetna, who's been very helpful in getting that created and used. The main developers for us, for Wikidata, are Andra Wagmeister and Sebastian Bergstaller, who's a postdoc in our group, also Elvira Matraka from the Disease Ontology group, and many other people that do less work but get more money. And the funding and support for our various projects come from here. So I will stop there, and we have a couple of minutes for questions if anybody's left. So thank you very much.
Thank you. On Biobranch, you mentioned students, and you also mentioned expert users. So, what sort of students, what sort of people do you expect to be most interested in Biobranch?
It's really tailored at the moment in its current incarnation for expert users. Okay, so people, the people that we have tested it on, were a breast cancer research group in Scripps that was, so we went and tested it with their lab. And the way they used it was they basically used it to go and look at the pathway that they cared about, that they worked on in their lab, and how it related to that prediction problem. So that, I think that's something that we see. So for example, that for them to ask a question like, given this gene, how predictive is it of this phenotype? And given this gene in this context, so in older patients, in tumors that are very large or very small, or in cases where the recurrence score is high or low and so forth, for them to be able to ask that question effectively right now without an application like this requires them to hire a bioinformatician. And so that is really the kind of group of people that we were aiming at. In constructing the tool, I really do think it could be repurposed very effectively in classes to teach about machine learning. But we haven't done that yet. We don't teach students very much here at Scripps. But I'd be really interested to see if anyone else could use it for that purpose. And so, I mean, really, it's really meant to open things up to people that are interested in this kind of data and playing with it. And that could be a large set of people. Okay?
Thank you.
Sure.
Are there any other questions for Ben? Okay. Well then, thank you, Ben, for the presentation. It was a great presentation and initiated great discussions, great questions. So thank you.
Okay. Thank you for being here.
Yeah. So, and that concludes our presentations for today. Thank you, everyone, for attending today's conference.
Thank you. Bye-bye.
Thank you.
Bye.
Thank you.